All right, this is going to be a third section of notes. We're still in section 1 of chapter 14, but we're going to try to review a couple of things first of all, and then we're going to move on and try to finish up all of our notes for chapter 14. So the first thing we have up here is a karyotype, which is just the chromosome listing of all of the genetic information for an organism. And so first of all, we want to look at what we can tell based on this particular karyotype. So the first thing we should be able to identify is that this does not have 23 pairs of chromosomes, which is normal for a human genetic information. And so we know that this individual is not human. So with it only having 20 total pairs, we know it's not human because we would have 23 pairs and 46 total. So a couple other things that we can figure out is that because there is an XY set for our sex chromosomes, we know that this individual is a male. And we can also figure out that there are 38 autosomal chromosomes. Autosomal just means the non-sex chromosomes. So 38 autosomal chromosomes. We have 20 total pairs of chromosomes, and we have 19 pairs of autosomal chromosomes. So that's just some of the generic information that we can figure out based on this karyotype. And so moving forward from here, we're going to run through a quick example problem looking at cystic fibrosis. And cystic fibrosis is just a recessive disorder in humans, and in the notes here we're going to end up having a little bit more information on those. But based on this cystic fibrosis, we can figure out the genotypes of these individuals, or most of these individuals shown on the pedigree. And so I'm going to go up to the board and work our way through that. First four individuals shown here, Arlene, Tom, Wilma, and Carla, all have cystic fibrosis, so we know they have all recessive alleles because that's the only combination of alleles they can have in order to get the genetic disorder. And so the next couple I'll put up will be the heterozygous individuals. Okay, we know all of those for sure because they are either producing offspring that have the disorder. So George produces both Tom and Wilma. He has to pass on that recessive allele. Since he does not have cystic fibrosis, we know that he has to be heterozygous. Same thing can be said for Michael. Since Anne passed on one recessive allele, Michael has to pass on the other to allow Carla to be heterozygous. The only other two individuals that we can determine are Daniel and Alan. Since Tom passed on recessive allele, we know they have to be heterozygous, and I'll fill in the rest. And so the remaining three, Sandra, Tina, and Christopher, all will be unknowns because we don't know for sure if they are heterozygous or if they are holding on to two dominant alleles. And the last problem that we're going to look at today is a blood type and Punnett square. And so for our first individual, a woman who is heterozygous for type A negative blood is going to be marrying a man who has type A positive B negative blood. And so we're going to figure out the possible blood types that will be created from this individual. So here are each of the parents. Got a little wild with the first A, but A negative, and it's heterozygous, so it has the type O allele in here as well. That's also going to be negative. And we're crossing that with a man who has A positive, B negative blood. So I'll fill in our possible offspring. And so you can see our resulting offspring will end up with 4 different possible genotypes. So our ratio would end up being 1 to 1 to 1 to 1. And then with our phenotypes, we'd have 2 individuals that have A positive blood, since positive is the dominant allele. These would both be A positive. We'd have 1 individual that is AB negative and 1 individual that is B negative. So we'd end up with a 2 to 1 to 1 ratio. All right, now we're going to run through a few things in the notes. So if you were in class today, you can just ignore the first couple of slides here, and obviously with the example problems that you already saw in class. So the first thing that we're going to look at is a few codominant alleles, or one specifically called sickle cell disease. So with this particular codominant disorder, the sickle cell ends up creating a blood cell that is a half moon shape. And so the reasoning for this we'll get to in just a second, but the purpose of blood cells is to carry oxygen. And when they have this sickle cell shape, it reduces their ability to carry oxygen. So because of that, the individual that has that disorder is less likely to be able to carry enough oxygen and can sometimes cause some problems with circulation and getting oxygen to the tissues that need them. And so it's more common in African Americans, and as we move forward in the notes, we'll eventually see why that might be. The reason that genetic disorders end up creating problems for individuals is because of the ability of that specific sequence of chromosome to be able to make a protein. And so for the first one, sickle cell disease, the protein involved is hemoglobin, which is what actually carries the oxygen in blood cells. And if the hemoglobin is not able to be created correctly and will not fold correctly, it will not be able to make or carry that oxygen, which is shown here. So here's a normal hemoglobin protein folded appropriately, and at the bottom we have a sickle cell hemoglobin, which does not fold the right way and does not allow for oxygen to be held by that cell. And so I'll come back to that in just a second. But the key thing is that proteins cannot be created correctly because there is something not right with the genetic information when they get either the 2 recessive alleles or in some cases with dominant disorders, they get the dominant combination. So the genetic information does not create proteins the right way, and we get, like the hemoglobin here, a protein that doesn't work correctly. And so we can see this is just the protein that is involved in a normal blood cell and then the sickle cell blood cell. Another disorder that we'll look at is cystic fibrosis, and this is a recessive disorder, so not quite like sickle cell disease, which was a codominant disorder. So with recessive, obviously we need 2 recessive alleles in order for this trait to become present in the offspring. And a couple of the problems associated with cystic fibrosis is the main thing is that there's a mucus buildup in the lungs. And so when that mucus builds up, it can cause pneumonia, it can cause some other problems with individuals that have that. And the only way to really combat cystic fibrosis at this point is to wear a backpack-like structure that shakes and tries to break up the mucus and allow it to be coughed up and get gotten out of the lungs. So there is some newer methods where you receive an injection of the protein that allows it to then break down that mucus, but that's still in the relatively early stages of development and not used quite as widespread as it may be in the future. And so the main thing that's causing the problems is that because of the 2 recessive alleles, it doesn't create a protein that allows for chloride ions to pass across the cell membrane. And so when those chloride ions start to build up, that's what creates the mucus, and then that builds up in the lungs. And so here we can see just basically what's happening, the buildup of mucus and other materials inside the bronchial tubes, and then ends up creating a lot of congestion and can create some infections and other things. Back to sickle cell disease for just a little bit. So when sickle cell blood cells are flowing through the capillaries, it is more likely that they get stacked up on top of each other and end up clotting because they do not flow as easily as a rounded normal blood cell. And so because of the change in DNA by the sickle cell disorder, the amino acid valine, which we talked a little bit about amino acids previously, ends up creating the hemoglobin to be less soluble, so it does not end up absorbing as much oxygen in the lungs and allowing that to then flow throughout the body. But one thing with sickle cell disease that kind of led to it being relatively common in African Americans is that in Africa, being heterozygous for sickle cell disease, which means half of your blood cells are normal and half of them are sickle shaped, because remember this is a codominant disorder, those individuals end up being resistant to malaria, which is a very common disease passed by mosquitoes in Africa. And so if we look at the figures here, on the left-hand side, it's a percentage of populations in Africa that have sickle cell disease or sickle cell disorder. And so you can see higher than 6% of the population is throughout the very same area that has a higher prevalence of malaria. And so you can see also 2 to 6% is the lighter pink color, and still kind of overlapping some of that area where malaria is more common. And so there's a direct tie to those two diseases in how the likelihood of that getting passed on. So having the heterozygous form, not being completely devastated by sickle cell disease, allows you to better survive through malaria. So because of those two the environmental condition along with the genetic disorder makes it more likely that that is able to be passed on to future generations rather than just eliminated from the population. All right, so this is going to be the last slide. And so when we're looking at human chromosomes, we know we have 46 chromosomes. We went over kind of the review of that as well, 23 pairs, 44 autosomal chromosomes, 46 total, 2 sex chromosomes. But of all of that genetic information that's in your body, only a very small percentage of it is actually going to make the make the genes that make you who you are. So it's only about 2% that create genes, which are the proteins and the other things in your body that create your hair color and your eye color and all the other things. So most of your DNA does not actually make proteins and doesn't actually do anything that we know of. So each gene consists of about 3,000 base pairs, and base pairs consist of adenine, thymine, cytosine, and guanine. And those are our nucleotides, which when we move forward to chapter 12 after spring break, we'll talk more about each of those nucleotides and how that makes up the DNA. But we will cover a little bit more about that later on in our next portion of notes. All right, so again, make sure that you are watching these video notes and getting down the information that you don't already know.